Good morning, everyone. Today, our topic of discussion is sterilization. Sterilization is a very important topic under the module of microbiology, and sterilization is very important for controlling the microbiological work and processes. Now, moving on to the detail of the sterilization, first of all, let me tell you what things will be covered in this section. Sterilization, importance of sterilization, methods, mainly the advantages and disadvantages, effects of sterilization in conclusion, and finally the future prospects. Now, before moving on to the more detailing of the sterilization, first of all, we will talk about the definition of sterilization. Sterilization is a process or killing or removing of bacteria and all other forms of living microorganisms, their spares from preparation. Essential concept in the preparation of sterilization. The sterilization process mainly aims at the preparation of a sterile pharmaceutical products, like the presence of any unwanted microorganisms or contamination in a particular sample will lead to spoiling or decay of that sample. So, sterilization is very important while we are conducting any microbial work or any work which requires proper aesthetic or proper aseptic conditions. What is the aim of sterilization? It provides that a product is safe and eliminates the possible introduction of any microbial contamination. Now, moving on to the next thing, that is importance of sterilization. Why sterilization is required for in different aspects of our life. First of all is medical sterilization. In the medical sciences, in the medical biotechnology mainly, there are many regions where the sterilization is very much important. So, first of all, it prevents the growth of disease. Suppose a medical tool or device is kept in an open space, then the chances of contamination or growing of any bacteria is very important, is very likely to take place when this phenomenon occurs. So, if sufficient amount of cleaning or disinfecting is not done, so there are chances that from that equipment many diseases may arise. This point is described in this section of your preventing the growth of diseases. Now, next thing is the prevent the spread of disease. Why, when disease is communicating, one thing is growth of disease and another is the spread of disease. That means growth is the initialization or starting of any disease, and spread is the when the infection is transferring from one person to another. Same way, if a patient is treated in an instrument which is not sterilized, that will be required to expose, the patient will be exposed to a lot of contamination, which will lead to the spreading of the disease from one person to another. Maybe the patient is not infected with a microbe, but when it comes in contact with a particular type of instrument which is not sterilized, they will bear the disease inside themselves. Now, prevention of double surgeries. What does it mean? It means that when you're using some equipments and if you're not using a sterilized equipment, and suppose and that sterilized equipment, when unsterilized equipment is used to do any surgery, then the microbes present on those equipment will be transferred into the body of the patient, and this will lead to starting of another surgery to remove those things. So by sterilization we can remove double surgeries also. Now, moving on to the next important thing, that is the method of sterilization. We have already talked that how, what is the importance. That means sterilization is very much important in conducting any uh, biological work or any chemical work which is involving some microorganisms, which is involving some organism which is very much vulnerable to the attack of some microorganisms. So, coming on to the method of sterilizations, there are mainly three methods: physical methods, chemical methods, and mechanical methods. Now, physical methods are mainly dry heat sterilization, moist heat sterilization, and sterilization by radiation. And chemical methods are gaseous sterilization, sterilization by disinfectant, and coming to the mechanical method, passing through bacteria proof filter. Okay, now what is happening? how these three types have been established on the basis of the method of your sterilization. The physical methods, that means physically you are removing the bacteria from the particular place, from the body of any equipment or any place where you want to remove the bacteria. Chemical method is when you are using some chemicals, when you are treating the place with some chemicals to destroy the particular organisms. Now next is your sterilization by mechanical method. In mechanical method also is somewhat related to physical method also. These two are related. So uh, what things need to be done here is that the mechanical methods is mainly the controlling the uh, contamination by some filter type of a material. Now coming to each of the categories separately. Physical methods is, I have already explained that there are three types under the physical method. The first one is your dry heat, then moist heat, and then radiation. What is the instrument used in dry heat? It is the oven. And what is the instrument mainly used in moist heat? Is the autoclave. Autoclave is a very important instrument used in the biological labs, and autoclave is a mandatory instrument required for any biological lab, because when you are working with some microbes, when you are working with some organisms or any media, you need to purify the media inside some instruments. So this autoclave is a very important equipment, which is almost like a mini pressure cooker, which is used as, not mini, I will tell you that as the volume is very high, so it can capacitate a huge amount of media inside it, and that can be sterilized. Okay. And radiation, gamma ray chamber, we all know that gamma rays are capable of killing some important microorganisms. So through radiation of this gamma ray, the all the microbes can be killed. Achha. Now coming in detail to each of the sterilizer, first of all is the dry heat sterilization. Here the oven has been used, but how the oven has been used? This is the diagram of an oven. You can all you know about this oven. Especially it is a design instrument, and which in this instrument it can be exposed to 160 degrees Celsius for 1 hour. Sorry, it is suitable for sterilization of a substance destroyed by moisture. As we have mentioned here that the heat sterilization can be done through dry heat and moist heat. So in dry heat, all the material which is destroyed by presence of any moisture has been heated, has been treated with dry heat sterilization. And what are the disadvantages? A disadvantage is that it requires a long time for sterilization, and the temperature required is also very high, because as you are heating it in a dry state, you have to raise the temperature very high, and the heating time is also very high. Now coming to the moisture sterilization. Moisture sterilization is mainly done through autoclave. Autoclave, as I have told you, see, it is look like a mini pressure cooker, but nowadays more complicated and sophisticated instruments are available, and the autoclave does not look like this. It looks like a bit complex structure, in which what happens, you are rising the temperature of this system to 121 degrees Celsius and 13 psi pressure, and for 15 minutes you are heating the substances which is present inside it. First of all, what happens when you operate an autoclave, you have to rise the temperature to a fixed amount, that is 121. Then when you have risen to that temperature, and then that temperature you have to heat the substances present for 15 minutes. Why 15 minutes? It has been seen that if you heat a substance at 121 degrees Celsius for 15 minutes, then all the microbes present in it dies out. Okay. So this is the advantages. What is the advantage? Advantage is that the microorganisms are killed most efficiently in lesser time due to high pressure saturated steam. Okay. And then disadvantage is what? The disadvantage is that suppose an instrument, suppose a material which is very much thermolabile, that means at high temperature it cannot stand, it disintegrates into some other things, or it get deteriorated. So we cannot treat the, or we cannot use the autoclave for sterilizing this type of material, because when we are rising the temperature to 121 degrees Celsius, the material present inside the autoclave totally deteriorates and melts or changes into a different substance, so making the process very difficult. Now coming to the radiation, uh, the sorry, the sterilization by radiation is, it involves two techniques. One is alteration of chemicals lead to form of new compounds, and vital structure like nuclear protein are destroyed, killing the microorganism. That means through gamma radiation we can destroy the nuclear protein, which can be destroyed, and microorganism can be used. Example is cobalt 60. This is a very important compound used as for gamma radiation sterilization purpose, and generally it is a radioisotope. Then during and when what happens, in radioisotopes it disintegrates all the time and produces a ray, that is gamma ray, and which kills the protein, nuclear protein of a particular cell. And by the way, throughout the process it kills the organisms. So students, for today we will be discussing till the physical methods of sterilization. In the next class we will move on to the next slides, that is the chemical method of sterilization. Till then, thank you students.